A selectively permeable membrane is a biological membrane that allows certain molecules and ions to pass through, whilst other molecules cannot. The main factor that determines this is size, although lipid solubility and electrical charge can also influence whether molecules can pass through the membrane or not.
All cells have selectively permeable membranes through which substances exit and enter, possessing microscopic pores that determine what can or cannot enter the cell, as shown in this animation. In order for a cell to function, it must interact with its environment, controlling and regulating the movement of substances in and out of the cell through the membrane. In both plant and animal cells, this is the role of the selectively permeable membrane.
Here the membrane exhibits selective permeability, allowing the smaller molecules to pass through but inhibiting the movement of the larger molecules. In order to explain this concept more clearly, we have likened the nature of a cell with a selectively permeable membrane to a jail. Here, the jail complex itself represents the cell. Around this jail complex is the fence which represents the selectively permeable membrane. The molecules that are allowed to pass in and out of the jail are the prison guards, similar to the molecules that are able to pass through the membrane. The prison guards are able to move in and out freely as they have a security pass allowing them to do so, similar to a molecule being able to pass through the membrane as they are small enough to do so.
The prisoners are not able to exit the complex, much as some molecules are not able to pass through the membrane. The inability of the prisoners to get out of the jail because they do not have a pass represents the molecules that are too big to pass through the pores of the membrane. However, in cells there are substances that are not able to pass through the membrane on their own, and require a carrier protein to do so. These substances are demonstrated by the inability of civilians to enter and exit a jail by their own free will, but are able to do so with the presence of a jail warden.
This is how a jail represents a cell with a selectively permeable membrane.
The aim of our experiment was to investigate how molecular size affects the movement of substances across the selectively permeable membrane of an egg.
We hypothesized that substances with a large molecular size would not be able to move across the selectively permeable membrane of an egg.
There were a number of variables that needed to be controlled in order to ensure validity. These variables were the eggs, which were taken from the same carton, so were of the same type and age, as well as being of similar size. The concentration of substances stayed constant, as all substances were saturated in solution. The eggs were left in the solutions for the same period of time. The eggs were kept next to each other, thus keeping the environment constant.
The independent variable was the substance the egg was immersed in, as determined by its molecular size. These substances were starch solution, iodine solution, and water. The dependent variable for this experiment was the weight of the egg over time.
The eggs were placed in vinegar and left for 2 days, in order to dissolve the shells of the egg and expose the selectively permeable membrane. After this, the starch and iodine solutions were made, in order to be used as the treated groups for the experiment, with pure water then being used as a control. A glass was filled with a cup of the starch solution and 1 egg was placed in it. At the same time, another glass was filled with a cup of water as a control and an egg was also placed in it. They were then left for 5 hours in order to allow the substances to move across the selectively permeable membrane, and each hour were weighed to provide a steady progression of results.
The egg in the starch solution was then transferred into the iodine solution for another period of 5 hours, again being weighed hourly. After the 5 hours, the egg was examined to determine whether there had been a reaction between starch and iodine, as indicated by a change of color to purple or black. The process was then repeated in order to ensure reliability of the experiment. Finally, the results were collated, tabulated, and graphed. As you can see, the egg that has been immersed firstly in starch solution and then in iodine solution has not changed color as a result of a reaction between starch and iodine. This indicates that the starch molecule was not able to cross the membrane.
The weight of the egg in the starch solution, a treated group, was seen to go down, indicating the inability of the starch molecules to move across the membrane. Comparatively, it was seen that the egg immersed in water, the control, gained weight due to the water moving across the membrane into the egg. Finally, the egg in the iodine solution, the second treated group, also gained weight, showing that the iodine molecules were small enough to move across the membrane.
This graph shows the progression of weight increase and decrease of the eggs over 5 hours. It shows that the water was able to move across the membrane faster than the iodine, as facilitated by aquaporins in the membrane.
Since the substance with the largest molecules, the starch solution, was not able to pass through the membrane, and the substance with the second largest molecules, the iodine solution, pass through at a slower rate than the water, it can be concluded that molecular size directly affects the movement of substances across a selectively permeable membrane.